Hello everyone, my name is Virus Musfira and I am from UiTM Cawangan, Sarawak. Today I will be presenting our research titled Functional Group Analysis and Adsorption Efficiency of Untreated and Treated Spent Coffee Grounds in Crystal Violet Removal. Of course, this work would have not been possible without my wonderful teammates. Introducing Chemist Dr. Abu Yani, our leader, Technologist Dr. Siti Kartina, and myself, Virus Musfira. Let's start with the problem we wanted to solve. This is the structure of crystal violet, a bright synthetic dye widely used in textile, printing, and laboratory work. It looks harmless, but when released into rivers and lakes, it doesn't break down easily. It is toxic to aquatic life and potentially harmful to humans. The challenge? Getting it out of wastewater effectively and affordably. Now, there are existing methods such as chemical precipitation, membrane filtration. They work, but they are costly, energy-intensive, and often create second pollutants. So we ask ourselves, could we remove crystal violet using a greener, low-cost approach? Our answer came from something found in almost every kitchen and cafes, which is spent coffee grounds. They are free, abundant, and chemically, they have natural functional groups that can bind pollutants. So, instead of sending them to landfill, why not give them a second life as a water purifier? Our study had two main goals. First, to use FTIR spectroscopy to identify the functional groups in untreated and thermally treated spent coffee grounds, and second, to compare their ability to remove crystal violet dye from water at different dosages. We approached this in 3 steps. This is the overview of our methodology. First is the preparation and treatment of the coffee grounds. Second is characterization using FTIR spectroscopy. And the third one is batch adsorption studies. We started by collecting spent coffee grounds, washing them thoroughly and oven drying at 105 degrees Celsius for 24 hours. Then, we sieved them to a particle size of 850 micrometers. For the treated batch, we applied thermal treatment at 300 degrees Celsius for 2 hours in a muffled furnace. The untreated batch stayed exactly as prepared. Next, we analyzed both samples using FTIR, scanning from 4,000 to 600 per centimeter. We were looking for hydroxyl, carbonyl, alkene, carbon oxygen, and carbon nitrogen groups. These functional groups can bind dye molecules through hydrogen bonding, pi-pi interactions, and electrostatic attraction. In the absorption test, we prepared a 100 mg per liter crystal violet solution. We tested adsorbent dosages from 2 to 20 gram per liter, shaking them for 3 hours at room temperature. After filtering, we measured the remaining dye using ultraviolet spectrophotometer at 590.5 nanometer. Next, we proceed to the results obtained. Our FTIR spectra confirmed that both untreated and treated coffee grounds had the functional groups we were looking for. These functional groups play a role in binding crystal violet, which explains why coffee grounds are so effective as a biosorbent. Thermal treatment made some peaks more intense, suggesting stronger functional groups. But, as we'll see later, that didn't necessarily mean better performance. In the adsorption study, for untreated spent coffee grounds, dye removal went from 92.41% to 99.25% as we increased the dosage from 2 gram per liter to 12 gram per liter, and then it became constant. The maximum absorption capacity was 231 mg per gram. Interestingly, higher adsorption may reduce efficiency per gram, likely because some active sites were left unused. For treated spent coffee grounds, Removal stayed around 92% across all dosages with a maximum adsorption capacity of 230.7 mg per gram. So, while thermal treatment improves certain functional groups, it may have reduced porosity or blocked access to some active sites, slightly lowering performance compared to untreated spent coffee grounds. To wrap up, both untreated and treated spent coffee grounds work well for crystal violet removal. Untreated spent coffee grounds perform slightly better and is cheaper to prepare. FTIR confirmed multiple functional groups contributing to the adsorption of the crystal violet. This makes spent coffee grounds a sustainable, low-cost option for dye removal from wastewater. Our next step is to test spent coffee grounds with other dyes and contaminants and explore scaling this for real wastewater treatment plants. These are the references that we use in this study. On behalf of our team, thank you for your attention and thank you for watching. We hope this has given you a fresh perspective on how something as simple as coffee waste can make a big impact.